Ray, I have a question for you. I want to move on to, of course, data collection, which is a major topic of underlying everything we're talking about here. As you're collecting and interpreting early process and product data, that's going to guide scale up and later phase development. What are the strategies that you're using to ensure that that data is meaningful? What are the best practices and the actual strategies that you and your team use?
So I think one of the most important things that a lot of people don't necessarily start, a lot of companies don't necessarily start with, is to invest in your analytics early. Oftentimes when you are a lean company, the analytics that you base everything on are you have your release specs, and those are the assays that are run. But then oftentimes you're going to miss a lot of information, so that when you are later and you're looking at a potentially marketable therapeutic, you don't have a lot of the information that you could have gathered, and now it will be that much harder to make changes. As you get later, the FDA will require a much more comprehensive understanding of what your changes may be impacting from a product quality perspective. So when you invest in analytics early, beyond your release specs, I think it will allow you to gain a much greater understanding. I also think, where, again, this requires a bit more effort from a lot of earlier stage companies, is to monitor your product and process very closely, capture not as much all of the data that is there, and look at it regularly. What you might think is data that means nothing, there's a high chance that it could mean nothing, but there's also a small chance that it could mean something. And it could be critical. So capturing that and understanding it and analyzing it are incredibly important. And then one of the things that is also of paramount importance is, again, this is especially with autologous cell therapy, is to develop a cross-functional team to help characterize beyond CMC, because it is all patient-derived material, and there are things like prior treatments and translational aspects to understand. You want to have full understanding of the product that comes in, how it works when it goes out, so that you're not in sort of a CMC bubble where you think that your yields are very good or whatever it may be, but all of a sudden, maybe what you think is good is not efficacious from the patient treatment perspective. And on the same end, what your clinical operations team with the starting leukapheresis might think is a perfectly fine collection, you might find out, in fact, you can't, it does not grow as well because their prior therapy was a platinum-based therapy that didn't have a long enough washout time. So I think a lot of that is really important when you're looking at the future.
Great, thank you. I want to address a question that came in from Andrea, and she is asking, can you speak to decisions related to early consideration of larger scale shipping and distribution anticipated upon commercialization, including shipping of patient starting material for autologous therapies? And so, Ray,
Yeah, absolutely. So I think it's especially the patient starting material is a uniquely hard problem when it comes to autologous cell therapies. And I think, again, what that is starting the work early, even if that doesn't necessarily mean, putting out the capital that you need to do validation studies with your shipper, but you start with things like mock shipments to make sure that your lane validations that work out, and that your clinical site is capable of performing everything that they need to. And with that, it is of utmost importance to speak to your courier and your shipper. They might be one and the same, to understand how you might best scale your shipments. Autologous cell therapies, the way it works is oftentimes one patient, one batch, one shipment. So understanding the complexities of that, and potentially setting up, for autologous cell therapies, Oftentimes you want to freeze it to begin with. So setting up either a very robust supply chain for fresh blood material, or to set up a network that can help you cryopreserve locally, or to work with the clinical sites to standardize at the clinical site your cryopreservation process, so there isn't too much variability from site to site in how you freeze your material.
Thanks, Ray. I appreciate it. Before we move on, I see that Anshika has submitted a question, but the question didn't come all the way. It got part of it got deleted. So if you could please resubmit that, we would love to answer your question. While we wait for Anshika and others to submit questions, we'll move on. So Todd, I'd like to ask you, a large portion of Cell and Gene's audience is in fact small and emerging companies. And so let's talk to them a little bit about what are the practical steps to build that robust data strategy from the start to help them build and guide smarter decisions that lead to an eventual tech transfer. Does it look different from what the big guys are doing? And for small emerging companies, what's your best advice for them?
I wouldn't say it looks different per se, but you don't get as many chances on goal here. So you want to get it right from the start. Going back a little bit on one of Ray's earlier comments on analytics and our therapeutic space, these are the most complex therapeutics out there, and to really understand how the process moves and influences the final drug product and then the final drug product in translational. There's a lot of characterization assays that he alluded to and your spec assays, but I think probably our deep understanding will come from those characterization assays that ultimately have some mechanistic understanding. And some of these are challenging in vitro assays, et cetera. So back to the data part, that's a lot of analytical data. So coming to Allogene in the early days, one thing that I focused on was we have all this data and there's a lot of off-the-shelf tools for visualization, statistical analysis, et cetera. But the data isn't often amenable to those tools in a high throughput fashion. So the first area I would say to focus on is figure out how to structure the data, so it fits well with tools like Spotfire, Jumper, or similar. And this is often, it sounds easy, but you really have to get the whole audience, the whole company on board, because you can't name an attribute one thing in one data set, another thing, because then your analytics of that data falls apart on a grander scale. So I would say to sum that up, really put into place a robust data structure, and how are you going to manage that in a data lake, and what's that solution look like? And then building upon that, there's lots of good analytical tools to really do a high throughput look at those data. I would recommend, personally, as a small startup, there may be a temptation to build your own tools in Excel or others. Go to tried and true visualizations would be my recommendation. So you spend your effort on the science of developing cell therapy and not, coding in the early stages. I think this is where big pharma might go and do it on their own, because there might not be solutions for, now we're talking, terabytes of data, for example.
Great. Thank you, Todd.
Mm-hmm.
It looks like Anshika's question has come through, so thank you for resubmitting that. Could you please speak to moving away from fresh leukopak to frozen leukopak as a starting material? Ray, can I ask you to respond to that?
Yeah, absolutely. So obviously from a supply chain planning perspective, it is much easier to start with a cryopreserved material, because you don't necessarily, you don't have to plan around when the patient will be available. We all work in this industry. The complexities of leukapheresis collection are more than you probably anticipate. The patient could get caught in traffic on the way to their collection. So it might miss a plane and things, and all of these complexities. So transitioning to starting with cryopreserved material, there's lots of different considerations for that. So the first is, starting the process with the cryopreserved versus fresh. It really is, again, about understanding what the differences are. It's about doing the studies, it's about doing comparability, and ensuring that the cryopreserved material performs just as well as the fresh material does, or at least, it produces a product of similar product quality. Now, when it comes to how you do that, you can either freeze at your site of manufacture, or you can set up a sort of a network of local cryopreservation facilities that maintain a single, your single protocol, and that can, that has the benefits of reducing the complexities of your logistics. It increases the complexities of having more, what is essentially sort of small contract manufacturers. But again, it's preferable to be able to do that. Or even, if you want to take it a step further, is working with the clinical sites themselves who oftentimes have these capabilities, either to help them standardize to whatever process that you choose, or studying to make sure that your process that your process is robust enough to handle whatever starting material ends up coming through. But a lot of the industry starts with frozen material, and I think it's just ensuring that the same product quality that comes out is the same.